Behind your sternum, in the space between your lungs and your heart, sits an organ most people have never thought about since their last biology class, if they thought about it then. At birth, it weighed approximately 15 grams. At puberty, it peaked at around 35 grams, the size of a large strawberry. By the time you were 25, it had begun shrinking. By 40, it was mostly fat. By 65, it is approximately 85% adipose tissue, with roughly 15% functional remnants still producing, at a fraction of its original rate, the cells that stand between you and every pathogen, every cancer cell, every internal mutation your body has encountered since you were born. Thymus is the organ nobody talks about. It is also the organ that built you, specifically the version of you that can fight back. What it did, how it did it, and what its disappearance means for the body running on what it left behind. That is what follows. There is a selection process inside the thymus with a graduation rate of approximately 2%. There is a gene called AIRE that causes the thymus to simulate your entire body in miniature, presenting T cells with molecular samples from organs the developing immune cell will never visit during training. And there is a clinical trial from 2019 that showed the involuted thymus can regenerate in older adults, that the organ presumed gone can be partially woken. All of it comes later. Start with what the thymus actually is. Because nothing about the immune system makes sense without understanding the school that trained it. Thymus is a primary lymphoid organ, one of only two in the body, the other being bone marrow. It sits in the anterior mediastinum, divided into two lobes, each containing an outer cortex and an inner medulla, packed with specialized epithelial cells and developing T lymphocytes. T in T cell stands for thymus-derived. Every T cell in your body, every one of the billions circulating through your blood and patrolling your tissues right now, originated as a progenitor cell in the bone marrow that migrated to the thymus and underwent training there. Training is the most rigorous selection process in all of biology. The curriculum is molecular. The examination is pass or die, not metaphorically, but literally. T cells that fail are killed by apoptosis, their cellular contents recycled, their failed receptors destroyed. The graduation rate is approximately 2%. 98% of all T cells that enter the thymus die there. What the 2% that survive carry away, what they learned in the thymus and will carry for the rest of their lives, is the most important thing the adaptive immune system knows, the difference between self and everything that is not self. In 1961, a 26-year-old researcher at the Chester Beatty Research Institute in London removed the thymus from newborn mice and discovered what that organ was for, by observing what happened when it was gone. Jacques Miller's thymusless mice developed normally by external appearance. They ate, they moved, they grew. From the outside, nothing was wrong. Then their immune systems were challenged. They could not reject skin grafts from genetically different donors. The immune response that normally destroys foreign tissue was absent. They were susceptible to infections that normal mice handled easily. They failed to mount antibody responses that require T-cell cooperation. Miller had demonstrated, by removing it, that the thymus was the organ responsible for generating the cellular arm of adaptive immunity. Context matters. In 1961, immunology did not understand how the body distinguished self from non-self. The thymus was considered a vestigial organ, an evolutionary leftover with no clear function in the adult body. Anatomists noted that it shrank after puberty and assumed this confirmed its irrelevance. Miller's experiment reversed that assumption entirely. The organ they had dismissed as vestigial was running the selection program that made adaptive immunity possible. The shrinkage after puberty was not evidence of irrelevance. It was the beginning of a decline whose consequences would play out across the second half of every human life. He was 26. He received the Lasker Award in 2019 at 85, 58 years after the discovery. The recognition is often described as the most important immunological finding of the 20th century, and one of the longest delays between discovery and major award in the history of biomedical science. Now, before a T cell can learn anything in the thymus, it must build the tool it will use to recognize the world. and the way it builds that tool is the most extraordinary molecular engineering in all of biology. Every T cell needs a receptor, a protein complex on its surface that can recognize and bind to a specific molecular fragment called an antigen, presented by the body's own cells on MHC molecules. The challenge the immune system faces is this: it must be able to recognize pathogens it has never encountered, mutations that have never occurred, molecular configurations that do not yet exist. It cannot wait to see the threat and then design a receptor. It must have the receptor ready before the threat arrives. Solution is random assembly. The VDJ recombination system, driven by two enzymes, RAG1 and RAG2, randomly cuts and rejoins gene segments from three separate gene families on the chromosome, generating T-cell receptor diversity through combinatorial assembly. Each developing T-cell in the thymus independently runs this molecular lottery, producing a receptor with a binding specificity that is effectively unique, different from every other T-cell in the body, different from every T-cell that has ever existed. A random receptor is as likely to attack the body it lives in as to defend it. The thymus is the filter that catches the ones that would kill you. Number of distinct T-cell receptor configurations this system can theoretically generate exceeds 10 to the 18th power, 1 quintillion. The number of atoms in a grain of sand is approximately 10 to the 19th. Your immune system can generate nearly as many distinct antigen recognition configurations as there are atoms in a grain of sand, from a set of gene segments that fit on a single chromosome. Diversity is not encoded in the genome. It is generated by randomly combining fragments in ways that produce sequences the genome never contained. The immune system is not reading from a library. It is improvising, and the improvisations number in the quintillions. This is what makes the immune system fundamentally different from every other organ system in the body. The heart pumps with the same mechanism it was born with. The liver metabolizes using enzymes encoded directly in the genome. The brain processes information through circuits that were laid down during development. The immune system generates its recognition capacity from scratch, through random molecular assembly, producing tools that no genetic program specified. It is the only organ system that creates its own functional diversity through a stochastic process, rolling the molecular dice quintillions of times and then testing every result against the body it must protect. Two filters test every receptor the lottery produces, applied in sequence, and between them, they eliminate 98% of everything. First filter, positive selection, runs in the thymic cortex. A newly generated T cell with its random receptor encounters thymic epithelial cells displaying MHC molecules, the protein complexes that every cell in your body uses to present molecular fragments to the immune system. For a T cell to function, its receptor must be able to interact with at least one of your own MHC variants. If the receptor cannot bind to any MHC molecule, if the random assembly produced a configuration that is structurally incompatible with the body's own antigen-presenting language, the T cell is useless. It receives no survival signal. Within days, it dies by apoptosis. Approximately 70% of T cells fail positive selection. They drew a receptor in the molecular lottery that cannot read the body's language. They are eliminated, not because they are dangerous, but because they are useless. A T cell that cannot interact with MHC molecules will never detect an antigen, no matter how perfectly its receptor matches a pathogen. It would circulate for years, consuming resources, occupying space in the repertoire, and contributing nothing to immune surveillance. The thymus eliminates it before it wastes a lifetime of resources on a cell that cannot do its job. The thymus is already down to 30% of its starting class after the first filter. 70% gone. Not for being harmful, but for being incompatible with the body they were supposed to protect. Second filter. Negative selection is, if anything, more important, and the mechanism behind it is one of the most remarkable discoveries in immunology. Surviving T cells move to the thymic medulla, where they encounter medullary thymic epithelial cells expressing a protein called AIRE, autoimmune regulator. What air does is extraordinary. It drives the expression of tissue-specific proteins from organs throughout the body in thymic medulla cells. Insulin, normally produced only by the pancreas. Thyroglobulin, normally produced only by the thyroid. Retinal proteins. Liver enzymes. Lung surfactant proteins. Proteins from tissues the developing T cell has never visited and will not visit during its time in the thymus. Air causes the thymus to simulate the entire body in miniature, to present developing T cells with molecular samples from every tissue they will later patrol, expressed solely so that developing T cells can be tested against them before release. No other organ in the body performs this function. No peripheral mechanism replicates it with comparable fidelity. The test is this: T cells whose receptors bind too strongly to any of these self-proteins. Cells that would, if released into circulation, recognize the body's own tissues as targets, are eliminated. Destroyed by apoptosis in the medulla. This is negative selection. The T cells that survive both filters, that can read the body's MHC language but are not attracted to the body's own molecular identity, are the approximately 2% that graduate. They are the only ones released into the peripheral blood. They are your adaptive immune system. That mechanism, AR, expressing proteins from organs the T cell has never visited. The thymus running a full-body molecular exam on every cell it produces, is the finding that stopped me when I first encountered it. Because it means the thymus is not merely filtering, it is teaching each developing immune cell the complete molecular identity of the body. It will spend its lifetime patrolling. And the organ running this program has been shrinking since puberty. Well, and here's where the failure mode becomes specific. Because every autoimmune disease is a failure of this filter. Rheumatoid arthritis, type 1 diabetes, Hashimoto's thyroiditis. Each one represents a T cell that should have been eliminated in the thymic medulla and was not. A cell that drew a receptor in the molecular lottery survived positive selection because the receptor could read MHC and should have been caught by negative selection because the receptor also recognized a self-protein, but was not caught. It was released. It survived. It eventually encountered the self-protein, its receptor matched, joint tissue, pancreatic beta cells, thyroid proteins, and attacked. Attack is not indiscriminate. It is targeted with the molecular precision the immune system uses against pathogens, because the mechanism is identical. The autoreactive T cell is doing exactly what it was trained to do: recognize a specific molecular pattern and destroy the cell presenting it. The error is not in the execution. The error is in the selection. The thymus should have identified the receptor as dangerous and eliminated the cell before release. It missed. Mechanism is confirmed by the genetics. Mutations in the ARE gene cause a condition called autoimmune polyendocrineopathy syndrome, type 1, APS1, in which negative selection is globally impaired and multiple autoimmune diseases develop simultaneously. The thymus, when AR functions correctly, is the most important protector against autoimmunity in the body. When AR fails, the consequences are precisely what the mechanism predicts. Autoreactive T cells escape in numbers, the peripheral tolerance mechanisms cannot contain, and the body attacks itself across multiple organ systems. When it works correctly, the thymus prevents this. The thymus that is declining, involuting, replacing functional tissue with fat, is running this filter with progressively less capacity and less fidelity. Think about what this means for the person over 60 whose thymus is 85% adipose tissue. The remaining 15% is still running the selection program, still producing T cells, still testing them against self-proteins through AIR-mediated presentation. But the volume is reduced, the throughput is a fraction of what it was at 12. And the question of whether negative selection at reduced capacity allows a slightly higher rate of autoreactive T cell escape. Whether the accumulation of autoimmune conditions with age is partly a consequence of declining thymic filtration quality is one that the immunology of aging is actively investigating. The correlation is consistent. The mechanism is plausible. The data is still being built. Now, the involution itself. Because the disappearance of the thymus follows a specific timeline driven by specific hormones, and the consequences play out across the entire second half of life. Thymic involution begins at puberty. The mechanism is hormonal. Estrogen and androgens both drive the replacement of functional thymic tissue with adipose tissue. The timing is not coincidental. It is directly linked to the rise in sex hormone levels that puberty produces. Animal studies confirm this. Castration, removing the source of sex hormones, produces partial thymic regeneration even in aged animals. The sex hormones that launch reproductive maturity simultaneously begin dismantling the organ that built the immune system. Evolutionary logic is debated. One hypothesis: the thymus generates the T-cell repertoire during development, and once a sufficient repertoire is established, the organ's continued function provides diminishing returns relative to the metabolic cost of maintaining it. Peripheral T-cell survival and homeostatic proliferation. The mechanisms that maintain existing T-cell clones through cell division take over from central thymic output as the primary means of immune repertoire maintenance. Handoff from thymic production to peripheral maintenance works adequately in young adults whose repertoire was recently and fully stocked. It begins to fail in older adults as peripheral T-cell renewal cannot compensate for the accumulated loss of thymic output over decades. The problem is not merely numerical. Each time a peripheral T-cell divides to maintain the population, its telomeres shorten slightly. After enough divisions, the cell reaches replicative senescence. It can no longer divide, and it shifts into a pro-inflammatory state that contributes to the chronic, low-grade inflammation characteristic of aging. The peripheral maintenance program that was supposed to preserve the repertoire is simultaneously degrading it. Each maintenance division producing slightly less functional cells with slightly shorter lifespans and slightly more inflammatory profiles. Immune system you are running at 65 was largely generated by an organ that was at peak function when you were 12. T-cell repertoire patrolling your body today was shaped by selections that occurred in a thymus that has been declining for 50 years. Pathogens you encounter at 65 are being matched against a repertoire that the organ responsible for updating and diversifying it is no longer maintaining at meaningful rates. Decline is measurable with a specific molecular marker. T-cell receptor excision circles. TRECs are small circular DNA fragments produced as byproducts of VDJ recombination during receptor assembly. They are generated once during the recombination event, and they are not replicated when the cell divides. Each cell division dilutes them by half. A T-cell fresh from the thymus carries a full complement of TRECs. A T-cell that has divided 20 times in the periphery carries almost none. TREC levels in the blood are therefore a direct readout of how much recent thymic output is entering the peripheral pool. TREC levels decline continuously with age. By 65, thymic output measured by TRECs is approximately 95% lower than at 20. The naive T-cells, cells with fresh receptors that have never encountered their antigen, are being produced, but at rates too low to maintain the diversity the peripheral pool requires. Consequence is immunosenescence, the progressive narrowing and aging of the immune repertoire. The peripheral T-cell pool becomes dominated by memory T-cells that have already encountered their antigens and clonally expanded to fill the available immune space. The system is increasingly specialized, better at responding to previously encountered threats, progressively worse at handling novel ones. Practical consequence is specific, and the person over 65 has lived it. The cold that lasts 2 weeks at 65 when it lasted 5 days at 35. Partly because the naive T-cell response to the specific viral strain is slower and smaller than it would have been with a fuller repertoire. The shingles reactivation that occurs decades after the original chickenpox infection, the varicella-zoster virus, was held in latency by T-cell surveillance, and as the repertoire narrows with age, the surveillance weakens, and the virus reactivates from the dorsal root ganglia where it has been hiding since childhood. The pneumonia that hospitalizes at 70 when it would have been managed at home at 40. The immune response mounted by a depleted repertoire with fewer naive cells available for primary response. None of these are failures of the immune system in the sense that something broke. They are consequences of a system running on a repertoire that is no longer being replenished by the organ that built it. You see, this is why the influenza vaccine is less effective in older adults. Not primarily because the vaccine is different, because the naive T-cell pool that would respond to novel vaccine antigens is depleted. The thymus that would have been replenishing it has been declining for decades. The vaccine presents its antigen to an immune system that has fewer fresh cells available to mount a primary response. The reduced efficacy is not a failure of the vaccine. It is a consequence of thymic involution measured at the cellular level. And then, one finding from stress research that connects the thymus to the cortisol physiology the series has covered across multiple scripts. The speed of the connection is startling. Hans Selye, the researcher who defined the modern concept of biological stress, documented in the 1930s that rats subjected to acute stressors showed measurable thymic shrinkage within hours. Not days, hours. The mechanism is glucocorticoid mediated. Cortisol binds to glucocorticoid receptors on developing thymocytes and triggers apoptosis. The same programmed cell death that eliminates failing T cells during normal selection now triggered prematurely by stress hormones in cells that had not yet completed their training. A single acute stress event producing a significant cortisol spike can reduce thymic cellularity by measurable percentages within 24 hours. The thymus recovers after the stressor is removed. The epithelial scaffold remains, and new progenitor cells from the bone marrow repopulate it. But periods of chronic stress, the sustained elevated cortisol of prolonged difficulty, grief, caregiving, or chronic illness, produce sustained thymic suppression that reduces both the volume of T-cell output and potentially the quality of selection during that period. Clinical observation that autoimmune flares follow bereavement, trauma, or periods of severe chronic stress has a potential thymic mechanism. Cortisol-driven impairment of negative selection during the stress period may allow autoreactive T cells to escape elimination that would have occurred under normal cortisol conditions. The mechanism is plausible and consistent with the known sensitivity of thymocytes to glucocorticoids. It has not been directly demonstrated in humans. But the thymus of a newborn mouse subjected to acute restraint stress shrinks by up to 50% in cellularity within 24 hours. The organ running your immune school is exquisitely sensitive to the stress chemistry you produce when your life is hard. That finding is the one that tied the thymus to everything else in this series, for me. Because it means the immune education that determines what your body can and cannot defend against is not running in isolation from your emotional and psychological state. The cortisol, the Greek script described, the cytokine-driven inflammation, the cortisol elevation of sustained emotional pain, is reaching the thymus. The stress the waking script described, the morning cortisol surge, the chronic elevation of poor sleep, is reaching the thymus. The organ that decides which T cells live and which T cells die is listening to the chemistry your life produces, now. The finding that changes the trajectory of the entire story. Because the thymus was presumed gone. In 2019, Gregory Feynman and colleagues published a clinical trial in Nature Aging that produced a result almost nobody in the field expected. The TRIUMPH trial, thymus regeneration, immunorestoration, and insulin mitigation, used a combination of recombinant human growth hormone, DHEA, and metformin in 9 men aged 51 to 65. MRI imaging before and after the 1-year intervention showed measurable increases in thymic volume, with adipose tissue replaced by functional-appearing thymic tissue. TREC levels increased, confirming that the regenerated tissue was not merely structural, but was functionally producing new T cells with freshly recombined receptors. The subjects also showed a reduction in epigenetic age, biological clock measurements based on DNA methylation patterns, suggesting their cells had become approximately 2.5 years younger on average during the 1-year trial. Sample was small, 9 subjects. No placebo control group. The finding was preliminary in every sense that matters for clinical medicine. But the direction of the result was consistent across all 9 subjects. Not one showed thymic shrinkage during the trial period, and the TREC increases confirmed functional output rather than merely structural expansion. The mechanism was consistent with earlier animal data. Growth hormone and IGF-1 drive thymic epithelial cell proliferation and reduce glucocorticoid-mediated thymocyte apoptosis. The hormonal signal that shrank the thymus can be partially countered by a different hormonal signal that expands it. Organ did not die. It went to sleep. Maintained in a quiescent state by the hormonal environment, the epithelial scaffold preserved beneath the adipose tissue, capable of partial reactivation when the hormonal signal changes. The strawberry that became fat did not lose its architecture, it lost its activation signal. Epigenetic age finding, the 2.5-year reversal, deserves separate attention because it suggests the regeneration is not merely local to the thymus. If restoring thymic output produces systemic changes in the epigenetic markers that track biological aging, the implication is that thymic decline is not just a consequence of aging, but a contributor to it. That the depleted immune repertoire is itself accelerating the biological processes that age the rest of the body. Restoring the thymus may not merely restore immune function, it may slow the aging that immune decline produces. The data is too preliminary to state this as established fact. It is not too preliminary to state it as the most interesting implication in current aging research. Multiple independent approaches, caloric restriction, specific cytokines like interleukin-7, mTOR inhibition through rapamycin, all produce thymic expansion in animal models, pointing at the same conclusion. Thymic involution is not an irreversible biological inevitability. It is a hormonally driven, potentially reversible process. The strawberry-sized organ that built your immune system and then shrank to almost nothing is still there, still structurally present, still containing the epithelial scaffold that could, if the signals change, resume production. And here is what all of this means for how the body ages. Because the thymus connects to every major vulnerability of the second half of life, decline in thymic output is one of the clearest mechanistic explanations for why older adults are more susceptible to infections, less responsive to vaccines, more prone to cancer, and more likely to develop autoimmune conditions as they age. Cancer surveillance deserves specific attention. Every day, cells in the body acquire mutations through normal DNA replication errors, oxidative damage, and environmental exposure. Most of these mutations are repaired, some are not. The mutated cells that escape DNA repair present altered proteins on their MHC molecules, molecular flags that mark them as different from normal self. T cells with receptors that recognize these altered proteins destroy the mutated cells before they can proliferate. This is immunosurveillance, the immune system's continuous scan for abnormality. And it depends on a diverse repertoire that can recognize novel molecular patterns. As the naive T-cell pool depletes with thymic involution, the repertoire's capacity to recognize novel tumor antigens narrows. The mutations that would have been caught by a fresh T-cell with precisely the right receptor are no longer being caught because that receptor configuration is no longer present in the depleted pool. The cancer that establishes at 70 may have been prevented at 30 by a T-cell the thymus produced, and that the peripheral maintenance program eventually lost. The immune system running at 65 is the system the thymus built and then left on peripheral maintenance alone for 40 years. It is not failing. It is aging on a trajectory set by an organ that began declining before you were old enough to notice. If thymic involution can be slowed, reversed, or compensated for, the immune consequences of aging change with it. The infections that kill older adults disproportionately, influenza, pneumonia, sepsis, are in significant part consequences of a depleted naive T-cell pool. The cancer surveillance failures that allow malignancies to establish in older bodies are partly failures of a repertoire that can no longer generate fresh cells against novel tumor antigens. The autoimmune conditions that accumulate with age may partly reflect declining quality of negative selection in the thymus running at 15% capacity. Thymus is not a footnote in the biology of aging, it is one of its primary mechanisms. Jacques Miller removed the thymus from newborn mice in 1961 and discovered what it was for by observing the collapse that followed. He was 26. The recognition arrived when he was 85. Between those two ages, the thymus in his own body ran the identical program it runs in every human body, built the immune system, stepped back, and let the body maintain what it left behind. The assumption for decades was that the stepping back was permanent. That involution was the thymus dying, that the organ was gone and the body was on its own. The assumption was wrong. The epithelial scaffold is still there, beneath the adipose tissue, in the anterior mediastinum where it has always been. The architecture that ran VDJ recombination, positive selection, negative selection, the 2% graduation program that built every T cell in your body, is structurally present. Hormonally suppressed, not destroyed. The thymus did not disappear. It was told to stop. The question the next decade of immunology will answer is whether it can be told to start again.